So is my screen visible?
Yes, sir.
Yes, sir.
Yeah, okay. So I think in the last class I started with the antiviral drugs, right? So we are in the fourth unit. So fourth unit is of anti-cancer drug molecule. Okay, so what are the mechanism of the most commonly used anti-cancer drugs? Then antiviral drugs. Okay, so what are the list of commonly used antiviral drug molecules and the molecular mechanism of antiviral drugs? Then antibiotic. So what are the very commonly used antibiotics for killing the bacteria, and what's the molecular mechanism of antibiotics, right? So I started with the antiviral drug molecules in the last class. So basically, all the antiviral drug molecules will have some resemblance with endogenous ligand molecule, thymidine. Okay, so that is what we discussed. So if you compare the structure of thymidine and IDURD, you can find very good structural resemblance between both, right? So basically we can classify the antiviral drug molecules into base modified or sugar modified. Okay, so if you take nucleoside. Okay, nucleotide is the building block of DNA. If you take nucleoside, which is a sugar plus the base, right? So if the modification is at sugar part, they are called as sugar modified, and if the modification is at base part, then those are called as base modified. Okay, very basic thing, right? So in this session, what I'm going to do is I'm going to specifically discuss about the antiviral drug molecules which are specifically used for human immunodeficiency virus (HIV). We'll see the molecular mechanism of few drug molecules. Okay. So this already I discussed, right? So we can classify the HIV drug molecules into different classes. One class of molecule is called as NRTIS. It stands for nucleoside reverse transcriptase inhibitor, right? So the main job of this nucleoside reverse transcriptase inhibitor, these are the molecules which goes and inhibit an enzyme called as reverse transcriptase, and the main function of this reverse transcriptase is conversion of RNA into DNA. Okay, so it converts the RNA back to DNA. That's the main function of the reverse transcriptase. So these are the set of inhibitors which goes and inhibits this reverse transcriptase enzyme. So what it does, it stop or prevent the conversion of the RNA into DNA. The RNA, the viral genetic material, cannot get reverse transcribed into the corresponding DNA molecule. That's the main function of this, right? So the molecular target for all the molecules which are listed here are the reverse transcriptase enzyme. Okay, so zidovudine, didonisin, stavudine, abacavir. So these are some of the very commonly used drug molecule that inhibits the reverse transcriptase enzyme. So coming to the molecular, okay, so these are the structures of the drug molecules which are listed, right? So at least you should remember the structure of, a very commonly used drug molecule, at least 2-3 structure you have to remember for the examination point, right? So structure of zidovudine is important, dianosine is important, right? So the structure of abacavir is important, right? So like that, it is not necessary to remember all the structure. That's not possible. So you can pick at least 2-3 structure which you feel like easy to remember. That's important for the examination point. Okay. So coming to the molecular action of the drug molecule zidovudine. Okay, so so HIV we have different types. Okay, so we have type 1, type 2, like that we have different types of HIV, right? So this specific molecule zidovudine, it's very specific for both type 1 and type 2. So it can act on both HIV type 1 and HIV type 2. So the molecular mechanism is, as I said, it targets the reverse transcriptase enzyme of human immunodeficiency virus. So that is considered as one of the very potential drug target, because if you prevent the conversion of RNA into DNA, So we can stop the complete replicative cycle of the human immunodeficiency virus. Okay, so this target is considered as one of the very important target. So this molecule perfectly inhibits the reverse transcriptase enzyme. So it acts as chain terminator in the reverse transcription reaction. Okay, so the chain elongation process will take place. So each nucleotide will come and join. Okay, to form the DNA molecule. So this molecule zidovudin, if this molecule goes and bind, so what exactly happen is that it acts as a terminator. Okay, the chain elongation process will not takes place because it lacks the OH group at the 3' position, which I already discussed in my previous lecture. So because of that, the chain elongation process will not takes place, which means that so uh, so 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 the chain elongation process will not takes place. So termination here will takes place. So it acts as chain termination in the reverse transcription reaction. So the molecular mechanism is almost similar for all the drug molecules. So initially this zidovudin will be in its pro-drug form, inactive form. So once if the molecule enter into the body with the help of some enzyme which is converted into the active metabolite. So here the triphosphate form of this zidovudin. So initially there are no phosphate group attached to zidovudin. So if you look at the structure of zidovudin, okay, so you cannot find any phosphate group in this structure. So this molecule as it is a prodrug, which is in inactive state. So once if you take this molecule inside the body, so we have a lot of enzymes which converts this zidovudin into its monophosphate, diphosphate, and triphosphate, which I already discussed in my previous lecture. The mechanism is same. So the triphosphate form of this zidovudin, it goes and incorporate in the viral DNA chain, and hence it causes the termination of the chain elongation process, right? So which means that it prevents that process. So transcription translation process are inhibited. So we cannot expect the virus to get reproduced. That's a molecular mechanism of this drug molecule. So so coming to the dose and mode of administration, so this molecule is preferred to orally administered at 600 mg per day, not as a single dose, right? So mostly they prefer to give 2 300 mg tablets daily, right? So that's a molecular mechanism of zidovudin. Okay, so here A is a T. Okay, so which is converted into its corresponding monophosphate with the help of kinase, then converted into diphosphate with the help of another kinase, and then converted into triphosphate with the help of another kinase. Then this triphosphate form of the drug molecule. So here you can see 3 phosphate groups which are attached. So the triphosphate form of this OH group is very important for the chain elongation. Okay, since this OH group is attached with other 3 phosphate group, so once if this molecule goes and incorporate in the DNA chain elongation process, okay, the next nucleotide cannot come and join because it lacks the 3' OH group. That is a molecular mechanism of this AZT. So the next molecule is danosine. This is also very very well used drug molecule, right? So coming to the activity spectrum of this molecule is very active for HIV HIV type 1 and HIV type 2. So for both the cases we can use this drug molecule. So mechanism of action, again the molecular target for this drug molecule is the same. So it acts exactly on HIV reverse transcriptase. The molecular mechanism of any antiviral drug molecule will be almost same. So since the main function of this molecule is it act as a chain terminator. So here also the phosphorylation takes place. So if you look at this structure of the drug molecule, okay, so as it is, the drug molecule is in its pro-drug form, does not have any phosphate group. So once if you take this drug molecule enzymatically, this sorry, enzymatically, this molecule is converted into metabolite. During that process, this OH group, right, so will be converted into attachment of phosphate will take place at this position. So, the 5' triphosphate form of the drug molecule is the most active form. So again, the mechanism is same. Okay, it go and attach to the 3' end of the viral DNA chain. So since it lacks the OH group at 3' position, the next nucleotide which cannot come and attach to that. So it's a chain terminator. Exactly, the mechanism is very similar to this, right? Any antiviral drug molecule, the mechanism is going to be almost very similar or common, right? So coming to mode of administration of this drug molecule, it is orally administered at 400 mg per day, or 200 mg tablet twice a day, or 200 mg tablets once a day, depending upon what's the condition of medical condition of the infection. So the another very important drug molecule which is very commonly used for HIV infection is zaltitabine. So the activity spectrum, again, this molecule also very potential for type 1 and type 2 HIV. So mechanism of action is same, molecule target is same. It targets HIV reverse transcriptase. Whatever I discussed for the previous two molecules, that is applicable here. So it acts as a chain terminator. Okay, it terminates the elongation process, DNA synthesis process. So it also phosphorylated form of the drug molecule is the active form. So as it is, this drug molecule is a prodrug, and it's metabolite. Okay, phosphorylated form of the drug molecule is the active form. So coming to the administration of this, this molecule is administered orally at 2.25 mg per day, 0.75 mg tablet for every 8 hours, right? So this is the dose regimen for this drug molecule. So so the another molecule is okay, so this is tonofovir disproportional. So this is another very successful molecule which is used against HIV. So this is also used for both type 1 and type 2 HIV here. So coming to the mechanism of action, so it is an oral prodrug. All the drug molecules which I discussed so far is an oral prodrug. Okay, so it targets the HIV reverse transcriptase or HBV reverse transcriptase. It's another enzyme, another virus, right? So the mechanism is same, act as a chain terminator. Okay, so initially it is its prodrug and converted into the phosphorylated state. So the diphos. Okay, so 3' end, it goes and binds to the 3' end of the viral DNA, and specifically this molecule also lacks the OH group at 3' position. So the next nucleotide cannot come and attach. So the chain termination will take place. The elongation process will be terminated. So coming to administration of this drug molecule, this molecule is orally administered 300 mg as a 300 mg tablet per day, right? So there's a molecular mechanism. So initially, the monophosphate is converted into diphosphate, converted into triphosphate form of the drug molecule, goes and incorporate in the DNA, right? So here the termination will take place. The next nucleotide cannot come and attach because this lacks the 3' OH group. Okay, that's a common mechanism for all the drug molecules which I discussed so far, right? So coming to the next class of drug molecule, non-nucleoside reverse transcriptase. So far what we discussed is nucleotide reverse transcriptase inhibitor. So this is a non-nucleotide reverse transcriptase inhibitors. These are example which I mentioned in my previous lecture itself, right? So the specialty of this molecule is, so you have to remember at least one or two structure of non-nucleoside reverse transcriptase inhibitor, right? So better you can go with the first molecule and the third molecule. So nevirapine is very successful molecule which act only against the HIV type 1. So we cannot use this for HIV type 2. The general principle of non-nucleoside reverse transcriptase inhibitor. So these are the set of molecules which won't go and bind to the active binding site. Okay, they prefer for the allosteric binding site. Meaning is that, so these are the molecules which goes and bind to the allosteric binding site of the enzyme and alters the shape structure of the active site of the enzyme. Okay, it's not directly binding to the active site of the enzyme. So mechanism of action is target at an allosteric pocket of HIV1 reverse transcriptase. Very important. So the target is reverse transcriptase. Okay, but the binding pocket is not the active binding pocket. The binding pocket here is it is allosteric binding pocket. This molecule bind to the allosteric binding pocket and alter the structure of the active site. Okay, so it disturb kind of the original activity of the enzyme. So coming to the dose mode of administration, so this molecule also preferred to be administered as oral drug molecule at the concentration of 200 mg per day for the first 14 days. So 200 mg tablet per day, then 400 mg per day, two daily 200 mg tablets. So these are called as a dose regimen. You might studied this in your medicinal chemistry, right? So I don't want to go in detail, right? So based on the pharmacokinetic properties, this regimen is the best dose regimen for this nevirapine. Okay, so these are the set of protease inhibitors, right? So I already discussed all this, right? So I just wanted to show the structure of protease inhibitor. So again, it is not necessary to remember the structure of all the molecules, but at least you know one or two very important drug molecules, right? So very commonly used drug molecule, you have to remember the structure, right? So indinavir is very successful drug molecule. Lopinavir is another successful drug molecule, right? So you have to remember the name of at least you know one or two very often used drug molecule which is very successful in the market, right? So coming to saquinavir, so HIV protease inhibitor. So the activity spectrum of this molecule is it's very promising for both HIV type 1 and HIV type 2. So mechanism of action of this drug molecule is it's a transition state inhibitor. Okay, so in the third unit, so I'll be giving the recorded session of for the third unit, complete third unit, right? So in which I discussed about what is meant by transition state inhibitor. So the substrate should go to the transition state before it get converted into the product. Okay, so this is the substrate, this is the transition state of the substrate, and this is the product, right? So the structure of substrate in its ground state and in its transition state will differ, right? So if you mimic any inhibitor, if you mimic any inhibitor that mimics the structure of the substrate molecule in its transition state, those inhibitors are what are called as transition state inhibitor, right? So no need to worry about this. I'll give the recorded session, right? So you can easily understand what is meant by transition state inhibitor. So mechanism of action of this molecule is it is a transition state inhibitor, right? So which is a peptidomimetic inhibitor, right? So protease, the substrate for protease is some protein molecule, right? So peptidomimetic means the inhibitor is also is a kind of peptide. It's not protein, it's a kind of peptide that mimics the substrate molecule. So coming to the administration of this molecule, so it is administered orally at 3600 mg per day. So the dose regimen is 200 mg soft gelatin capsule 3 times a day. Okay, again, this is fixed based on the pharmacokinetic property of this saquinavir. Okay, so this is structure of HIV protease. Okay, so if you remember, many times you might visualize this molecule in your laboratory session. So say for example 1-HSG is an example for HIV protease. You can see it is a homodimer molecule. So this is the indinavir molecule, okay, which is a very potential HIV protease inhibitor. So coming to discovery of influenza virus, right? So far what I discussed is human immunodeficiency virus, right? So now we move to another type of virus called as influenza virus. So influenza, which is considered as very dangerous, right? So very dangerous virus. What is some of the basic points related to influenza virus? They belongs to Myxoviridae family, and has single-stranded segmented RNA as the genome. So for this type of virus, influenza virus, the genetic material is RNA, it is not DNA. So we can subdivide this influenza virus into three distinct types. So type A influenza virus, type B influenza virus, and type C influenza virus. So among this type A, type B, type C, so influenza A virus is considered as very important virus, because those are the viruses which causes very significant diseases in human, right? So not the B type or C type. So this influenza A virus can be further classified based on the antigenic properties of two envelope associated surface glycoproteins. One is the hemagglutinin, another one is the neuraminidase. So these are the glycoproteins which are I'll be showing the image of this virus in some other slide, right? So say this is the virus, and in the surface, you'll be having many protein molecules, surface protein molecule. So there are two important surface protein molecules in this influenza virus. One is called as hemagglutinin, represented by Ha. Another one is called as another one is say this is another type of uh, surface protein called as neuraminidase, which is represented as Na, right? So again, this Ha and Na are of different types. So if you take Ha, so the we have H1 type, H2, H3 up to H16. And if you take Na, so neuraminidase, we have different types of neuraminidase. So say N1, N2 up to N9. So any of the combination, if H1 and N1 is present in the virus, then it should be called as, that virus should be called as H1N1 virus. Okay. So if H2 and N9 is present, then we should call this as H2N9 virus. Okay. So if H2 and say N1 is present, then H2N1 virus. Okay. This is the way how we can name this virus, right? So the take home point is like we have two important types of surface protein on this virus. One is called as hemagglutinin, and another one is called as neuraminidase. Okay. So impact of influenza virus, right? So just like this pandemic situation, right? Coronavirus, right? So influenza also outbreak sometimes, right? So it has great impact on human health. So high toll in morbidity and mortality with 500,000 death worldwide, right? So you had to think, right? So now we are talking about coronavirus, very dangerous, right? So even much more dangerous is influenza virus, millions of death recorded because of the infection of influenza, right? So this virus is kind of human to human transmissible. Not all the viruses are human to human transmissible. Be some viruses, it can transmit from some other animals to human. Only some type of viruses can be transmitted from human to human. Okay, so this is considered as one of the virus which is very easily transmitted from one human to another human, and highly pathogenic, right? So just like coronavirus, this is a highly pathogenic. So we can remember this number, right? So easily in 2009, if you remember, right? So if you take the news reported on 2009, there was an outbreak of infection, novel influenza A virus, just like what the situation that we are having now, right? So like that very new virus was evolved. They termed it as H1N1, because the hemagglutinin type present in that virus is of H1 type, and the neuraminidase present in that virus is of N1 type, right? So there's several deaths reported, right? So this is very important, right? So for the examination point, this is very important. So on the left hand side, this side, we have the overall structure of this virus, not the very detailed structure, but whatever is important for understanding the molecular mechanism. So we have, right? So this is a representation of influenza A virus, showing the viral surface glycoprotein. So as I said, we have different types of glycoproteins which are labeled here. So one is called as HA. Okay, so this is a glycoprotein molecule. Okay, so here it is labeled. So this is called as hemagglutinin, and we have another type of glycoprotein called as Na, which is called as neuraminidase. Okay, so we have another important type of ion channel called as M2, right? So this Na, Ha, and this M2. Okay, Na and Ha are a glycoprotein molecule. M2 is an ion channel, right? So these three are considered as the most important drug target for influenza virus. So right hand side, we have the mechanism of replication, right? So how it replicate. So here this is the virus particle, right? So as we know that whenever the virus has to make a infection to any other animal system, right? So any other living organism, first it should come in contact, right? So it should be in contact with the host. Okay, host is the system whose machinery will be used well by the virus for its reproduction. So what happened, this is the structure, right? So the entire structure is represented like this: the virus particle which approaches the host cell, right? So this is the extracellular region. So this region is the extracellular region, and this region is the intracellular region. So the virus particle slowly attached to the extracellular part. Okay, so then the virus, it should enter. It should either enter into the cell, or it should inject its genetic material inside the cell. So in this case, this virus particle undergo a physical phenomenon called as endocytosis. So I hope you might study about the details of endocytosis in your molecular cell biology, right? So with the help of endocytosis, the entire virus particle is now entered into the host cell. So you have now assume that the virus particle which is entered into the host cell, okay, which is ready to make use of uh, the host machinery for its reproduction. So here there's a kind of envelope here. Okay, so what happens is, so because of the entry of this, so more amount of H plus ion, right? So the acidity of the environment will be increased, right? So it increases the acidity with the help of this ion channel, M2 ion channel, right? So main job of M2 ion channel is, it allow the H plus ion to enter into the cell, right? So once if this H plus ion concentration increases, we can say that it is more acidic, right? So the lysis process will take place. This envelope will be broken down. Okay, this envelope will be broken down. So once if it is broken down, so here we can see that genetic material will be slowly released. Okay, this is the viral genetic material that is released inside the host cell. So now this viral material is attached to the host, DNA, right? So where the replication process takes place. So also the mRNA is synthesized here. Here you can see, yes, so within the nucleus, right? So with the help of the host machinery, so whatever virus particles are needed for the formation of new virus, those are synthesized within this region, and slowly that is released to the cytoplasm from the nucleus. So whatever is required, right? So assembly is taking place here. Whatever proteins are needed, whatever genetic material is needed, all are assembled together. So budding process takes place, then the virus particle is released, right? So this is the general mechanism of the replication process or reproduction of this influenza virus. So what are the major drug targets? The first major drug target is so this is considered as a major drug target. So here for the binding, right? So here first the virus particle should come and in contact with the host cell, right? So then slowly it undergoes the endocytosis process and releases its genetic material, right? So what happens is that the host cell, it has a receptor called as sialidase. Okay, so the glycoproteins which are present in the virus makes interacts with sialidase, right? So because of that interaction, the virus particle is able to enter into the host cell. So these are the drug molecules which inhibits zanamivir and oseltamivir, very successful, very promising drug molecule that inhibits the binding of the virus particle, attachment of the virus particle, interaction of this glycoproteins present in the virus particle with the sialidase of the host. Okay, so the first is, we can say this is an entry level inhibition which will prevent the endocytosis of the virus to the cytoplasm, right? The second major drug target is that this neuraminidase and hemagglutinin. Okay, so those are the things which are actually involved here. I don't want to go in detail, right? So the another important drug target is inhibition of this ion channel, because if we successfully inhibit the ion channel, M2 ion channel, so the protons, H plus ions, cannot get enter into the cytoplasm. So the acidity of the environment will not increase, so the lysis process will not take place. So the genetic material of the virus cannot be released in the cytoplasm, right? So this is considered as the molecular mechanism of reproduction of that influenza virus. So coming to what are the anti-influenza drug molecules which are commonly used, amantadine, rimantadine. These are very common antiviral drugs, right? So zanamivir, oseltamivir, ribavirin, right? So H1N1, they have used this oseltamivir, zanamivir. These are very commonly used drug molecule, but there are cases in which the drug resistance reported for almost all the drug molecules, which indicate that if any new pandemic situation arises because of this influenza virus, so we may have a lack of drug molecule. We need some potential drug molecules, right? Still there are many companies which are working on influenza virus to come up with new drug molecules which are not resistant for the virus, right? So one of the major drawback of drug discovery for viruses, like the viruses, they can cleverly easily undergo some kind of changes that can resist the functioning of the antiviral drug molecules, right? So these are the structure of very commonly used anti-influenza virus compound, right? So again, you may not be able to remember the structure of all, at least, right? So one or two you have to remember, right? So amantadine, rimantadine, the very simple structure, oseltamivir, zanamivir, the very commonly used drug molecule, right? Okay. So coming to the molecular action of the specific drug molecules, amantadine. So the activity spectrum of this molecule is it is very specific for influenza A virus. So I already told that even though we have influenza A, B, C, right? The most important type, in human infection is the A type, right? So this molecule is very specific for influenza A virus. So if you have influenza B virus infection, or if you have influenza C virus infection, this molecule may not be potential. This is not a potential molecule. Okay. So mechanism of action is it blocks M2 ion channel. So amantadine is it blocks the M2 channel. Okay. So if I go back to the molecular mechanism, so this is the place where this molecule act, right? So the main function of M2 channel is that, so it allows the H plus ion to get enter, so that the acidity of the environment will be higher, right? So the lysis process, it supports the lysis of the virus particle, right? But if we inhibit this M2 channel with the help of amantadine, so we can stop the entry of the H plus ion. We can stop the increase in the acidity in that specific region. So hence we prevent the lysis of the virus particle. It cannot release the genetic material inside the cytoplasm. Okay. So that's it act as it blocks the M2 ion channel and thus prevents the passage of H plus ion that are required for necessary acidity to allow for decapsidation, right? So this process, right? So the lysis process is what is called as decapsidation. So which means that viral uncoating process, right? So we stop the viral uncoating process completely. So coming to the mode of administration of this drug molecule, so this molecule is orally administered at 200 mg per day, so two times 100 mg capsule. Again, this is fixed based on the ADMET property of the drug molecule and the pharmacogenomics of the person who is going to take this drug molecule, right? So coming to the second successful drug molecule for this influenza virus is rimantadine. So activity spectrum, again, this is also very specific for influenza A virus. So mechanism of action, it blocks the M2 ion channel, right? So it blocks, it's not entry level, right? So this molecule should enter into the cell and block specifically the M2 ion channel and prevents the passage of H plus. So the mechanism of rimantadine and the mechanism of amantadine are almost same, right? So if we inhibit, again, if we block the M2 ion channel, which prevents the acidity that is required for decapsidation, so the viral uncoating process will be inhibited. Viral uncoating process will not take place. So coming to the administration of this drug molecule, this molecule is orally administered at 300 mg per day. So it is preferred to give as a two dose, two times 150 mg. So coming to the another successful drug molecule for influenza virus is zanamivir. So interestingly, this is the first molecule that targets both influenza virus A and influenza virus B. For both the types, we can use this, right? But the previous two molecules we can use only for influenza virus A. So coming to the mechanism of action of zanamivir, uh, zanamivir and oseltamivir. The oseltamivir is another drug molecule, right? So both are very successful drug molecule. They are analogs of this salicylic acid. So as I told that, so for the entry of this virus particle, right? So the glycoproteins which are present in the virus should go and attach to acid which is present on the host cell called as salicylic acid. Okay, since zanamivir and oseltamivir are analogs of this salicylic acid, Okay, so instead of binding to the salic acid, so what they can do is they can go and bind to the salic acid and inhibit the attachment of virus particle to the host cell, right? So targeted at influenza viral neuramidase, okay, CLidase. So keep the virus trapped onto the surface of the cell, cannot release and cannot infect the other cell. So we can say this is an example for entry inhibition. Okay, so we are not allowing the virus to get enter into the cell. So we understand that the process by which the virus enter into the cell, we successfully perfectly inhibit the place which prevent the entry of the virus into the cell. So coming to administration of this molecule is preferred as inhalation. Very important. The mode of administration is inhalation. It is not oral. So you had to ask yourself a question: why this molecule is given as inhalation, right? So this is because based on the ADMT property of the drug molecule, so which is inhaled at a dose of 20 mg per day. Okay, so coming to oseltamivir, this also the mechanism is same, right? So very exactly the mechanism of zanamivir and oseltamivir are same. So just like zanamivir, oseltamivir also active against the influenza virus A and influenza virus B. So coming to the mechanism of action, zanamivir and oseltamivir. Okay, so same thing what I said, right? So it is analogs of salic acid, so which targets same thing, right? So administration is also there is a difference in administration, right? So rest of the thing, everything is same, molecular mechanism, everything is same, right? So previous case zanamivir is administered as inhalation, but in this case oseltamivir, it is not administered as inhalation, it is administered as oral drug molecule at 150 mg per day. So the proper drug regimen is 2 times 75 mg capsule every 12 hours for 5 days. So this is the drug regimen for this molecule. So coming to ribavirin, it is a successful antiviral drug molecule for influenza. So activity spectrum is like this molecule is can act for various viruses, not only for influenza, right? So this can act against the viruses which are having DNA as the genetic material or RNA as the genetic material. So the mechanism of action of this drug molecule is a target IMP dehydrogenase, which converts IMP to XMP, a key step in the de novo biosynthesis of GTP and DGTP. So we have to accept that GTP and DGTP are very crucial for the synthesis of DNA. Okay, and since this it prevents the conversion of IMT to XMP, right? So we can stop the production of GTP and DGTP. Okay, so coming to the administration of this molecule, it's orally administered at 800 to 1200 mg per day, right? So that's all about some of the basic concepts on antiviral drug design. Okay, so what are the commonly used antiviral drug molecules and what's the molecular mechanism of those antiviral drug molecules? So let me show you the syllabus, right? So okay, so we are in fourth unit, and I hope that I completed the antiviral drugs. Okay, anti-HIV compounds, nucleoside reverse transcriptase inhibitor, nucleotide reverse transcriptase inhibitor, non-nucleoside reverse transcriptase inhibitor, protease inhibitor, viral entry inhibitor, anti-HBV compounds, anti-herpes virus compound, anti-influenza virus compound. Okay, so I think this part is completely covered, right? So we have two more topics on the fourth unit. One is antibiotic another one is anti-cancer, right? So next class I'll be talking about antibiotics. Next to that I'll be talking about the anti-cancer agent. So any doubt up to this, I can answer. For the third unit I'll be giving the complete video lecture, right? So we won't have online session for the third unit, because almost everything you might already study in your medicinal chemistry and biochemistry, right? So I just wanted to give the recorded version of complete unit, right? So any doubt we can discuss now. So any doubt? So if no doubt, I just would like to wind up the session.
Sir,
Yeah.
there is this part in the
Okay, okay.
syllabus after anti-HBV
Uh,
compounds, anti-herpes virus. Did we discuss that?
Anti-herpes virus, I discussed in the previous session. Have you attended the previous session?
No, sir.
No,
Okay,
that's
sir.
the
I'll
problem.
just check.
Okay, I discussed anti-herpes. Okay. Anyway, I'll give the all the lecture tutorial. No issues. Okay. I just want you to encourage to read the textbook. Anyway, I'll be uploading all the recorded session. Okay.
Okay, sir.
Yeah. I'll give. So any doubt? So I got some question in chat. Learning the names of drug is enough. Okay. You know, say if you take anti-HIV drug molecule. Okay, so we have some 25 drug molecules. So definitely we cannot remember the structure of all the 25 drug molecules. So I strongly recommend you to remember the names of those 25 drug molecules, a dose regimen, okay, and the molecular target for each class. Okay, if I say non-nucleotide reverse transcriptase inhibitor, if I say protease inhibitor, if I say integrase inhibitor, if I say fusion inhibitor, at least for each class I should know one structure. Okay, for the examination point, so if the question is asked in essay question, essay type question, so you can make a tabular column with a name, what's the mode of action, everything. Okay, so for each class you can give one structure as a example. Is it fine? Yeah. So any more questions? So how many quizzes we completed?
4, sir.
Okay, 4 quizzes we completed. Maybe we will have one more quiz, 5 quizzes, and 3 more assignments I'll give. Okay,
Okay,
so
sir.
totally 8. Huh? So out of 8, we'll take the best 5. So 5 MCQ type quizzes and 3 assignment based quiz. Okay.
Okay, sir.
Okay,
Okay,
for
sir.
the date for the next quiz, I'll tell, right. So in a day or two, I'll decide and I'll tell the date for the next quiz, and also the assignment will be uploaded shortly, right. So maybe in 1 or 2 days, I'll upload the 3 assignments, right, and I'll give the due date for each assignment. You can submit before the due date. So any more questions?
No, sir.
Okay, then I think we can wind up the session. Okay, I think you have the project reviews now. You can prepare for that.
Thank you, sir.
Yeah. Okay. Yeah.